Hi, this is Amber Saunders, Camelia Thompson, Elizabeth Ferrer, and Kristen Drake, and today we will be investigating magnesium's effect on exercise performance due to its role in glucose availability. Magnesium has a lot of different functions in the body, including functions in energy metabolism, glycogen breakdown, which correlates to energy metabolism, nerve conduction, protein synthesis, cell growth, and muscle contraction. Magnesium may increase glucose availability and reduce lactate concentrations during exercise, thereby increasing performance. Exercise performance is highly dependent on the regulation and maintenance of magnesium homeostasis, and furthermore, a deficiency of magnesium leads to an impairment in exercise performance. Other signs of a magnesium deficiency include muscle weakness, nausea, and irritability. Magnesium is found in high concentrations in hepatocytes in the liver, and magnesium homeostasis is controlled by hormones, and those hormones move the magnesium in and outside of the cell. Magnesium supports and regulates specific liver metabolic functions and is highly compartmentalized within the hepatocytes and plays key roles in regulating enzymes, channels, and metabolic processes within each cell compartment and organelle. This diagram shows the dynamic scenario of how magnesium regulates glucose homeostasis and the utilization by regulating all these different enzymes and channels and processes. So in the cytoplasm, this is the cell membrane, so inside the cell membrane you have magnesium's effect on glucokinase and glycolytic enzymes and ATP utilization. And in the mitochondria, it regulates mitochondrial dehydrogenases and also pyruvate dehydrogenase via promoting the activity of pyruvate dehydrogenase phosphatase that dephosphorylates the enzyme to its active form. Then in the ER, in the endoplasmic reticulum, it regulates protein synthesis and the entry of G6P for oxidation by hexose 6-phosphate dehydrogenase to generate high levels of NADPH, which is used for other metabolic processes within the organelle and the cell itself, including fatty acid synthesis, cholesterol synthesis, and also the pentose shunt or the pentose phosphate pathway. So there have been many studies in rodents conducted to study magnesium's effect on glucose metabolism and exercise performance. This one in particular investigated the effects of magnesium sulfate on exercise performance and glucose metabolism in gerbils. So they used 24 gerbils and divided them into two groups. One was given a magnesium supplement and the other one was given a saline solution. And then the gerbils were forced to swim. Blood was drawn during and after exercise using an auto blood sampling system and a microdialysis analyzer to detect changes in glucose metabolism. This diagram here shows the effects of the magnesium sulfate supplement on plasma glucose levels. So here you'll see the administration of the magnesium sulfate supplement and how it affected glucose levels right away. This supplement was given 30 minutes prior to exercising, and then during exercising you can see the increase even more dramatically and the difference even greater between the two groups. So overall, the plasma glucose levels of the magnesium sulfate treated group during swimming and after 15 minutes of recovery were about 50 to 70 times greater than the control group. To further explain the differences between the two groups, you'll see from the basal levels that magnesium was increased, obviously, a lot more in the experimental group versus the control group. And then also you see a difference of glucose levels. You'll see that there's a difference of 70% in the between the control group and the experimental group. And then in regards to lactate, there is actually a 150% pretty much difference between the two groups, but in the opposite direction. So lactate was actually decreased in the magnesium supplemented group. So due to these differences in magnesium, glucose, and lactate in the blood, the supplemented group actually experienced a 71% significant increase in duration time compared with the control group. So overall, this study concluded that magnesium sulfate improved duration time of exercise by increasing glucose levels and decreasing lactate levels during swimming, thereby decreasing fatigue. So they accredited this increase in performance via increase of glucose availability due to magnesium's effect on the regulation of oxidative phosphorylation and the glycolysis pathway, and also magnesium's role in many rate-limiting enzymes. There is accumulating evidence of the relationship between magnesium and exercise performance due to magnesium's role in glucose metabolism. Research is still ongoing, but one day, dietitians may recommend magnesium supplementation to enhance exercise performance in human athletes. And furthermore, they may actually suggest increasing magnesium in diabetic patients because decreased levels of magnesium are being linked to insulin dysfunction. So overall, magnesium may have a magnificent future for its role in glucose metabolism.